Thank you, Simon, and good afternoon, everybody. My name is Dale Atty. I'm Chief Executive of Orla Protein Technologies, and really today I'd just like to, I was kind of thinking about what people would be interested in, and I guess you probably have questions like, what is METRC? How did the funding help? Is it worthwhile? What do you want out of it? So I've tried to kind of go through that from the Orla perspective, and then Gwen's going to talk about more interesting stuff and the science that we did in our project. And this isn't going to work on this side. So Orla is a nanobiotech company that spun out of Newcastle University in 2002. So we've been going a little while now. Our core technology is around proteins. We manufacture recombinant proteins. And particularly we have applications, commercial applications for those proteins for surface applications. And these have been exploited in a whole variety of areas, from surface analytical surfaces, biochips, through to cell biology. And we also have a joint venture with a large Japanese wireless communications company developing handheld diagnostic devices, which we've already heard a little bit about today. But today we're going to talk really about cell biology and cell biology applications of Orla technology. And our core technology involves putting recombinant proteins onto surfaces, and our unique USP is that we can create very, very structured and highly engineered surfaces, which give us many benefits for the different applications of the technology. So in effect, what we have is a toolkit, a nano engineering toolkit, which allows us to specifically engineer these biological surfaces for many different applications in life sciences and beyond into bioelectronics and other device platforms. So in the cell area, why are we interested in the cell biology area? Well, from a commercial perspective, it is quite an attractive commercial opportunity. And the culture of cell biology, the culture of cells, regenerative medicine, etc., is a huge and growing area. And products in this area can span everything from research tools through to large-scale and industrial scale-up and cell biology, cell manufacture. And within that, Orla is particularly focused on the kind of tools and surface applications with a view to industrial scale. And that is a particularly attractive market niche, which you can pick up these market research reports, and there's all sorts of numbers in there. But by any measure, it's quite a large and growing market opportunity. So that's why we're interested from a commercial perspective. However, there are many challenges for small SMEs in this area. Okay, we've got some great technology, we think, but there are so many challenges that we face in actually commercializing the technology, and we've heard a little bit about that from different speakers today. Now, one of the key barriers in this particular space, in the cell area, where protein scientists, protein technology, we don't have access to a wide variety of cell types and cell culture facilities. We need to generate valid data on our products and technologies, and access to that expertise and those facilities is actually quite difficult and often very expensive for a small company. So that's a one challenge where funding schemes like METRC can really help us a great deal. So the METRC project did allow us to address some of those challenges in this particular niche of interest to us. Gwen and I actually met prior to the METRC project. I think we met originally at a N3CR meeting, which is about reduction in use of animals in experimentation. And we identified a lot of common interests and synergies, but the usual story, okay, we've got lots of great ideas, but how are we actually doing things? Because we haven't got any money, they haven't got any money, we want to do something, what do we do? So around that time, METRC was launched, and we applied for one of the early METRC projects in the first round of METRC funding. So that did enable us to get an initial project underway. So that project was very specific and aimed at developing culture materials for bone cells, which is the expertise of Gwen's group here in Sheffield. And the objective there was to develop surfaces and culture materials that would act as mimics for the in vivo culture of these cells, and ultimately reduce the need for the use of animals in testing of this type. So Orla had already developed a number of early product opportunities in this space, and we have a collaboration with a rather large glass manufacturer, Lemon Sheet Glass from Japan. And we'd already developed a couple of 2D and 3D approaches, but we need to gain expertise in different areas of cell biology using these products, and that was the opportunity that the METRC project provided us with. So, very briefly, what were the outcomes from my side, from the commercial side in Orla, as a result of this project work on project funding? So, firstly, next slide. Clearly, we've established a relationship. As I said, we'd already met and worked out there were quite a number of things that we'd like to do. What the funding allowed us to do was to actually kick that off with a project, and that's always the best way to start, actually get some kind of project going. And Gwen will tell you all about the technical details of what we've learned from that project. And what that gives to us as a company is validation in a particular niche that we're looking at. So we need that academic rigor to apply to the products, the actual real data using real cells, and with a particular focus on the key areas that we're interested in. So we've got that very much from this type of project. Of course, added to that is the validation that comes from actually publishing those results. And as a result of this project, we do have some poster presentations, and we're working at the moment on the release of a new academic publication as a direct result of this project. So this is all great stuff as we are launching products in this area. You know, much of the work in this area is still coming out of the academic environment, so we need this validation to persuade people that these products are worthwhile. And obviously we'd like this to lead on to new things, and it already has resulted in some potential new funding opportunities for this particular area of our activity, and some of those I can't say too much about at the moment because they're still in the early stages, but certainly Orla has just taken part in a framework 7 application, which was somewhat related to some of the work that we've done here, and that has been successfully funded through Newcastle University. So those early seeds and the work that we've done on this project have already led to further applications for funding, and we hope that this work is now going to continue on. And we've learned a lot from this about where we should focus that next activity. So that's my little bit over. We'll get to the interesting things now about what we actually did. This is the second time you've seen this picture of the Centre for Life. So what Elaine didn't tell you is, so the only downside about METRC is they've helped Quantum DX move into my empire of the Centre for Life. So my plans for world domination have been slightly tempered by the fact that Elaine's company's now moved in next door. No, it's very nice to see you over there. Move along. Okay, I'll hand over to Gwen.
Yeah. The later book. Okay, thanks very much. Yeah, so as Dale said, we met at the MC3R meeting. I'm interested in bone tissue engineering, but particularly in the use of tissue engineering as alternatives to animal research. It's quite underfunded area at the moment, so it's something we're trying to break into and raise awareness of. So we work on musculoskeletal tissue engineering, in particular the interaction between the cells and the matrix, including the proteins in the matrix, which is where Orla, of course, comes in. And we're also very interested in mechanobiology, how mechanical forces affect how the cells behave. And this is some of our work on how fluid flow affects cell behaviour, and on 3D scaffolds where we see the cells into the scaffolds and we look at how they behave in 3D in relation to various forces. So, and why are we so interested in bone? Well, bone tissue loss is a major healthcare issue, and it's becoming more and more important with the issue of ageing and the elderly population and trying to keep people mobile. And there's problems with hip implants that fracture, or they have mismatch between the bone and the implant, so you get loosening of the implant, they need to be revised, this costs a lot of money. There's other places where metal is not suitable and people may use a bone graft, but then you have the issue of where can you get the bone from, will that cause disease transmission. There are some biomaterials available to replace bone, but most of these don't have the mechanical properties of bone, they're very brittle. So one option is tissue engineering. Can you encourage cells to make bone instead? Certainly at least to aid these kind of material implants, or even to eventually replace them. But the kind of materials we want to use and the kind of drugs we want to use so far are always tested on animals. And while we're making 3D bone, if that 3D bone, even if it's not implantable, maybe because of disease risk or cancer risk or something like that, can't we use these 3D structures to at least test our products and make better products in that way that haven't got to use lots of animals to test them? Especially with bone, the issue is that an animal's mobility is obviously very different from human mobility, so the mechanical forces are very different, the bone structure is very different, because most animals are not living as long, they don't have the same bone structure as humans. So we need a better model so we can accelerate creation of these materials. Next, please. So the kind of clinical applications are in diseases you're probably very familiar with, osteoporosis and osteoarthritis. Also there are links between bone and these big diseases that are becoming more and more of an issue, obesity and diabetes. And this graph is just to show that this bright green arthritis musculoskeletal has a bigger effect on limiting activity in older people, even than heart disease, which is the one we hear the most about. So musculoskeletal is very important financially to the health services. Next. Now, why is it difficult to grow bone? Why can't we just grow some bone cells in a lab, make some bone, and then stick it into a person, or test some drugs on it? Well, the problem is that bone structure is very, very complex nano and macro structure, and it's very difficult to replicate because it depends on the proteins that the cells are attached to, and it depends on the mechanical forces the bone experiences. So until you put those two things into your in vitro assay, it won't be the same as your in vivo. So we have the nanostructure of collagen, the nanostructure of the mineral, the way that it's piled up in layers, the way that this is arranged into the tube. And a critical part is the adhesion proteins. We're not going to show you in this diagram how to add them, but between the collagen and the mineral are very specific adhesion proteins that affect this structure. Next, please. So the aim of the collaboration was to produce a culture product where we could grow an in vitro bone-like matrix that really better represented bone than what's done at the moment, where cells are just grown on plastic, and that's not taken very far. The next step is going to animals very quickly. So we needed to find the right coatings, the right conditions. We started off with the 2D surfaces that Dale talked about, but quite quickly wanted to move up to 3D, because until bone cells in 3D, they're not really mimicking the environment properly. And I'd just like to mention that most of this work was done by the postdoc that we managed to employ with METRC funding, Katie Smith, and by a student that's employed by EPSRC funding, but we've used METRC to fund all of his project consumables, and a master's student who worked in parallel with our PhD student. So these are the oral proteins that we're interested in, because they're adhesion proteins that help bone cells adhere to their matrix, or because they're known to act as growth factors and affect the differentiation of bone cells. And if we think about tissue engineering, we want to think about taking immature cells and pushing them towards bone cells. It's very difficult to make bone cells that are extracted from bone to proliferate in the laboratory. Next, please. So we did some work with Orla-coated discs. I'm just showing this to be honest, because this actually wasn't so successful, unfortunately. So, of course, whenever you fund science, you're not going to fund everything successful. And this is because of issues we've had with serum-free media that are produced by other companies. So this wasn't necessarily something that we did wrong. It just happens that the right kind of serum-free media that we needed for this cell type was not available. This is quite a new cell type we're using, which is a bone precursor developed by this company, but not for use with this media or these surfaces. So this is something where we have to optimise this combination. But just to show there were some effects of the different adhesion factors we checked, but it was very variable, and we haven't managed to overcome this issue of the media on these 2D experiments. So that's ongoing work. Next, please. But in the meantime, we started the 3D work. This is Mossam's work. We've got a poster on about it as well if you'd like more detail. This bioreactor is shown in the poster. So here, what Mossam wanted to do is optimise the mechanical conditions we grow the cells in to get the right amount of matrix. So he looked at the flow rate, which is going from these pumps through the scaffold, which is in here, how often he put the flow on, and the number of bouts, the position of the scaffold, could it be horizontal or vertical, and some of these things we took from previous work we've done on other types of scaffolds, including, I see Tony in the audience, so I must thank Tony for all our preliminary work he's done with the polyurethane that he acquired for us. But now we're moving to the glass scaffolds, and we're changing some of the parameters to see which works best on the Orla glass. Next, please. So the first thing we showed was that we definitely do need some mechanical conditioning. Cells do like to grow on the Orla glass, of course, but a little bit of bioreactor conditioning, adding mechanical stimulus makes really nice dense cell culture. So where it's dark, we have live viable cells metabolizing. And when we look at collagen, which is the primary protein of bone, that's grown throughout the scaffold. And it's not very clear qualitatively, but I'll show you quantitative data that we've got more collagen in bone. What we're really pleased with is that we can see clearly into these scaffolds using confocal microscopy. A lot of commercially available scaffolds, it's very difficult to use under a microscope, and that limits the kind of questions you can ask on them. With this being glass scaffold with nice large pores, we've got beautiful pictures of the cell cytoskeleton, how it's attached to the glass, the nuclei, and of course we can start looking at specific proteins that we want to know if they've been up or down regulated by certain treatments. Next, please. So here's a little bit of the quantitative data. So this is telling us about the viability of our cells and their metabolic activity, and we'll see here the oscillating flow, which rather better mimics a walking cycle, and we could adjust that to really mimic a real walking cycle if we wanted to, have someone walk on a treadmill and input that data. At the moment it's just 1 Hertz, which is a typical walking pace. And that seems to better ensure cell survival compared to just one direction flow, which you might say mimics blood flow, but in bone we want to think about the activity as well as just a blood flow by a mimetic approach. And when it comes to collagen and calcium, definitely the oscillating flow seems to be a lot better in terms of improving the matrix production, the actual bone that we get in these scaffolds. Next, please. And we started to look at the coated scaffolds, so all that was done optimised with the plain scaffolds, and we are seeing very happily some effects of our coatings in terms of increasing the collagen and calcium production, and we're very happy to see that even in these commercial serum-free medias that we've had so much problem with, we are actually managing to get much better cell viability than producing collagen and calcium, and that's probably because in 3D cells behave very differently in terms of the interactions, and they maintain each other, and so hopefully we might be able to bypass some of the 2D work and concentrate on making a 3D matrix for these future tests. Next, please. So overall, we found that our serum-free media formulation is very specific, and we need further work to optimise those, which hopefully we'll be able to do in collaboration with some of these media formulation companies. But the cells do attach well and make bone matrix in the 3D Orla glass scaffolds, even in the serum-free media. We're optimising our bioreactor conditions, and the preliminary data indicates that we've got a condition that we can go ahead with, which is this 5 mils a minute of flow, oscillatory flow mimicking walking. And when we combine that with our coated scaffolds, we hope to optimise the coating to go to serum-free media, so then we can say, okay, if you buy this Orla scaffold, put your bone cells on it, you will have a good bone mimic that you can interrogate with your orthopaedic pharmaceuticals, and they may be eventually testing also materials with it. I looked up this, I put this word interrogatable into my PowerPoint, and it said it's not a word, but I'm sure it is. We want to interrogate our matrix. So that's as far as we've got so far. So obviously without the METRC funding, Mossam would have had to do a much easier, cheaper PhD that didn't have a consumables budget attached to him, probably sitting on a computer all day. And we wouldn't have had the opportunity to demonstrate that we're getting towards in vitro bone matrix. Bone is one of the really understudied tissues in terms of the field alternatives to animal experiments. and we really hope that we can break into that area and show that it's important to replace animals in this work and get a better understanding. So thank you very much for your attention.
Right, thank you very much. Does anyone have any questions?